Hello boys, welcome back to another interesting session of video tutorials, and this session is meant for class 12th biology students, and this is for 26th August 2020. And in this session, of course this is for 27th, right? 27th August 2020. And in this session we are going to discuss about a very interesting concept called translation. Like everything that happens in our body is in the form of code language. So the code language, as we discussed yesterday, will be in the form of a triplet code. And this is coming from DNA to mRNA. We call that step as transcription. And from transcription, when it has to come to mRNA, various procedures were there, we discussed about that. And from mRNA, the genetic information has to be converted into a chain of amino acids, finally forming a protein. So all our secrets are included in proteins. So this process of converting the coded language, which is hidden in mRNA, into a protein is called translation. So the code language which is there in DNA that comes to mRNA has to be decoded. And for decoding this information, genetic information, we require certain things. What are those? So first and foremost is mRNA, messenger RNA. Second, tRNA, transfer RNA or transportation RNA. Yesterday we discussed a lot about this structure of tRNA, amino acids site we said, DHU site we said, 4 arms we talked. And at the base there is something called anticodon we said. So keep remembering that. So first one is mRNA, second one is tRNA, and third ribosome should be there. Remember, ribosomes we say they are factories, protein producing factories we said. And then fourth, amino acids, because amino acids are monomers. When monomers join together, then only polymer will be formed. Here amino acids are monomers and proteins are polymers. And then to activate this amino acids, there should be an enzyme called aminoacyl synthetase. This is an enzyme, right? Anything that ends with ASC, I said, is an enzyme, aminoacyl synthetase. And then sixth one, peptidyl synthetase. Okay, peptide bond, remember? And of course, for all this process, we need to spend some energy. That's ATP. ATPs have to be spent, adenosine triphosphate, and GTP, guanine triphosphate. And then the catalyst should be there for every reaction, and the catalyst is Mg2+. So we need 9 things for translation to occur. So basically, translation, if someone asks you what is that, you have to say decoding the coded information carried by mRNA from DNA into protein. Synthesis of polypeptide chain on mRNA strand with the help of tRNA and ribosomes is called translation. Where is transcription taking place? DNA to mRNA, it's in the nucleus. But once mRNA is formed, it comes out from nucleus to cytoplasm. So translation takes place in cytoplasm. Okay. So again, in NEET exam, take care of that. So the process of translation takes place in 5 steps. So it requires 9 substances we listed. Now in 5 steps this translation takes place. First one, activation of amino acids. Second, attachment of activated amino acids to the tRNA. Remember, there is a site, one arm on the top. We said amino acid attached arm we said. So amino acids which are present in the cytoplasm, they have to be activated and made to travel, do some journey and get them attached to the tRNA, where on the top side, that is amino acid Armstrong we said yesterday. So attachment of activated amino acids to the tRNA. Then once it gets activated, remember the tRNA has got anticodons and mRNA has got codons. So initiation of polypeptide chain. Once the codon of mRNA, triplet code, matches with the triplet code of tRNA, the amino acid gets attached there, where on the top of the arm. And for this to happen, we talked about a particular triplet code called initiating codon. and I said it is easy to remember for exam, August, month of August, AUG. So that should be the initiation step of polypeptide chain. Then elongation. Once AUG starts off, then you got a second triplet code, third triplet code, fourth triplet code, so it continues. And as the tRNA starts traveling along with rRNA over the mRNA, you can see the elongation of polypeptide chain. It is something like, you have to imagine, I will show you the diagram, it is something like, railway platform. We'll talk about that. And then finally, anything that starts has to end somewhere. So termination of polypeptide. And when does this take place? I said 3 nonsense codons will be there, or senseless codons, or stop codons. And once in mRNA that stop codons will come, the anti-codon of tRNA and the stop codons will not match, and that is the signal for termination of the polypeptide chain, and protein synthesis gets stopped. So first one, as I said, activation of amino acids. The specific amino acid present in cytoplasm gets activated by specific enzyme. What is the enzyme? Just now I said amino acyl synthetase, and ATP is to be spent. It forms amino acyl adenylate enzyme complex. So amino acid plus ATP plus amino acyl synthetase in presence of Mg2 plus catalyst, I said amino acyl adenylate enzyme complex. Of course, ATP is becoming ADP and inorganic phosphate. Attachment of activated amino acids to the tRNA. Yesterday we were talking of this tRNA, clove shaped lavanga we say. So you can see the DHU loop of specific tRNA recognizes the activated amino acid according to its anticodon. So here you can see the anticodon, and DHU arm recognizes it, and then amino acid attachment is taking place at the site. The activated amino acid binds to third end of tRNA, 3' we said. Every thread has got 5' and 3' we said. So it results in the formation of amino acyl tRNA. So there is complementary base pairing. Yesterday we said 7 will be there, 7 plus 3, that is complementary base pairing. And down we have got anticodons. And now the ribosomal RNA, RNA. Now this is how the tRNA gets stuck with the tRNA synthetase. And there you can see on top amino acid. And there is something called active site. And once this tRNA synthetase takes the tRNA with the anticodons, now you can see the amino acid is getting activated, and the process ATP is spent, and the amino acid is getting stuck into that amino acid place, required place. And once that happens, now what is happening? The tRNA having anticodons, on top of it there is a high energy bond. tRNA is having very strong bond with the amino acid, and down you got mRNA, and always you said mRNA 53, then only activated. So the codons of mRNA should match with the anticodons of tRNA, then detachment of amino acids will start. And the initiation, as we said yesterday, DNA to mRNA is transcription, and mRNA to protein is translation. So you got basically mRNA, long thread, single thread, and then rRNA with 2 ribosomal units, one big one and small one. Yesterday I said 30S and 70S, and the initiating codon is August, AUG, and then the matching will be the methionine for AUG, specific amino acid, and the tRNA clove shaped with anticodons. Since initiating codon is AUG, matching should be here. If it is A, there it has to be uracil. Here it is U, there it is A, and here it is G, guanine, there it is C, cytosine. Then only the methionine comes and sits on the tRNA. So this is how the initiation takes place. So the ribosome binds to the mRNA at 5' end. The ribosome recognizes the initiating codon AUG on mRNA. The tRNA having anticodon UAC carries formal methionine acid to the site of initiating codon. It initiates polypeptide chain. This is the starting point of that. Now, once the initiation takes place, here I wanted to tell you, that you must imagine this mRNA as something like a platform, railway platform, and this tRNA and rRNA complex as something like a train. The bogies will run. So as the train runs, you can see the passengers will be coming out of the bogie and getting down, and like that you have to imagine the mRNA will be like a railway platform, and the rRNA tRNA combination, that complex, is something like a railway bogies. So bogie after the bogie, as the train runs, keeps on, and the passengers get inside. Here amino acids get inside, get stuck. So this is how elongation of the chain will take place. It is a linear growth of polypeptide chain on mRNA. You can see one after the other, as it moves, amino acids will be forming a chain. The charge of tRNA that carries specific amino acid enters the ribosome. It attaches to mRNA next to initiator codon with the help of anticodon. The peptide bond forms between these two amino acids are peptidyl synthetase. Again, there is a requirement of enzyme. As the peptide bond forms, the tRNA becomes uncharged. It leaves the ribosome. The ribosome moves codon by codon in the direction of 5 to 3. I said 53, you must remember. As the ribosome moves over mRNA, the coded information decodes into polypeptide chain. Again, see, you can see here the peptide bond between two amino acids, and of course you got a P site and A site that transport RNA, tRNA. You can see the matching of codons of mRNA with anticodons of tRNA. Once the matching takes place, then only the amino acid will be attached to the arm. Now you can see here, once the amino acid methionine gets released off, the RNA gets charged off, then you can see the formation of peptide bond between two amino acids. As the mRNA, the tRNA, ribosomal RNA moves over the platform of mRNA, many amino acids will keep on joining. You can see here further, as it moves, more and more amino acids are getting joined. This is how polypeptidal chain, you can see here, methionine, phenylalanine, glycine, leucine, methionine, you can see continuously chain, and this is how the continuous chain of amino acids take place. And finally, there has to be termination of the process. So the termination of polypeptide chain takes place due to the presence of nonsense codons or senseless codons. They are UAE, UAG, UGA. When terminator codon comes, it does not code for any amino acid. It leads to termination of polypeptide chain. The polypeptide chain synthesized releases out from ribosome. It undergoes folding to form a specific protein. The ribosome leaves the mRNA. Now you can see here the continuous chain of amino acids, and that should form a protein. And then stop codons are there that cannot match with the anticodons of tRNA. No amino acid will be joining there. That is the signal for termination. Now mRNA gets separated, rRNA subunits get separated, and finally we get what we wanted. That's new protein. So the main aim of the translation is protein synthesis. You can see here not many proteins like codon 1, codon 2, codon 3, mRNA, and there is a bond between the amino acids, and finally you get the protein that is wanted, and proteins are our secrets. So basically translation is mainly for protein synthesis. Now you need to combine both the transcription and translation, then you understand how the coded language from DNA enters into mRNA, from mRNA how the coded language will be decoded in the form of a protein. So keep on practicing.